Hello everyone, my name is Praveen and today I'm here to explain about the topic restriction enzymes. Restriction enzymes are also known as restriction endonucleases or molecular scalpels or molecular scissors. These are used to cut DNA at a specific location. Every restriction enzyme has its own recognition sequence and a target size. For example, let us consider a restriction enzyme that is known as EcoR1. This is the sequence of the EcoR1 which is known as the recognition sequence. This is known as the target site, a place at which the DNA can be cut into fragments. The EcoR1 can be cut into two fragments at its target site at two places. When the DNA sequence is cut by using a restriction enzyme into two fragments, the ends can be of two types. They are cohesive ends or blunt ends. Cohesive ends are also known as sticky, overhanging, or staggered ends. Blunt end is also known as flush end. Let us consider the same example, EcoR1. If the restriction enzyme cuts the EcoR1 at this site and the other strand at this side, then the both strands are not equal. So this is known as sticky end or cohesive end. If the restriction enzyme cut both the strands at the same side, then they both are equal and they are both blunt ends. As we know, every restriction enzyme has one recognition sequence and one target site. But a few restriction enzymes has their own recognition sequence and different target sites. And a few restriction enzymes have own recognition sequence and own target site. There are some exceptions in these restriction enzymes. They are known as neosisomers and isosisomers. The examples of neosisomers are SMA1 and XMA1. This is the recognition sequence of SMA1 and this is the recognition sequence of XMA1. Neosisomers have same recognition sequence but different target site. We can see the target sites of SMA1. They both are different but the sequence is same. Another exception is known as isosisomers. These isosisomers have same recognition sequence and same target site. Examples of isosisomers are SPH1 and BBU1. We can see the recognition sequence of SPH1. The target site is between GC and GC. So the target site is same for both. So these kind of exceptions are known as isosisomers. To find out the number of DNA fragments formed after cutting at a particular target site, we have one rule that is known as the n number rule. These are of two types depending upon whether the DNA is circular or linear. If the DNA is circular, we follow n rule. That means if we have n number of target sites, then the DNA fragments after cutting them will be of number n. If the DNA is linear in shape, then it follows n plus 1 rule. N is the number of target sites. That means if we have 4 target sites, then the number of DNA fragments formed are 4 plus 1 equal to 5. In this way, circular DNA follows n rule and linear DNA follows n plus 1 rule. To find out the number of fragments formed after cutting at the target site, we have to load the sample in agarose gel, and if we get two bands, that means there are two DNA fragments formed and we have one target site. In this way, we can determine the number of fragments formed by agarose gel electrophorese. Every restriction enzyme is determined by its star activity. Star activity is the number of bands that we expect and the yield we get. If we have less star activity, that means they are good restriction enzymes. If we have more star activity, that means they are bad restriction enzymes. The nomenclature of these enzymes was given by Smith and Nathan in 1972. The rules of nomenclature are that the first letter should be represented by the genus of the organism, and the second and third letters should be represented by the name of the species of the organism, and the fourth letter must be represented by the strain of the organism. At last, there will be a number that is known as the order of discovery. For example, let us consider a restriction enzyme named Hint3. Here, H is the genus of the organism, I and N are the species of organism, D is the strain of the organism, and 3 is the order of discovery. These are the rules used for naming any enzyme. There are various types of restriction enzymes. Generally, there are four types of restriction enzymes, but we study only three because the fourth type is considered as type 2S. So, there are three types of restriction enzymes. The type 1 restriction enzyme cuts 1000 base pairs away from the target site, and it is cheaper while compared to the type 2 and type 3. While coming to the type 2, it cuts exactly at the target site, and it is costlier while compared to the type 1 and type 3 also. The type 3 restriction enzymes cut 25 base pairs away from the target site, and it is cheaper than type 2. Using type 1 and type 3, we can remove unwanted DNA nucleotides. Here are some examples for cohesive and blunt and nucleo restriction enzymes. The examples of cohesive and restriction enzymes are XMA1, SPH1, BBU1, EcoR1, BAMH1, HIN3. The examples of blunt restriction enzymes are EcoR5 and SMA1. There are various applications of restriction enzymes, like analysis of DNA fragments, gene expression, study about cloning and recombinant DNA technology, DNA sequencing, and also site-directed mutagenesis. So this was all about restriction enzymes, its types and applications. I hope it was useful. Thank you.